Embark on the fungal ecology portion of this unit. Here we have a picture of a very beautiful fruiting body. Common name is the red-belted polypore. This is Fomitopsis pinicola, excuse me. And this organism is found often on conifer trees, fruiting on conifer trees. And also hardwoods, but more often conifers. And this is a particularly nice looking specimen. They can get a little funky looking as they grow, but here we can see this really nice red edge of this fungus, which gives it its name. And of course, underneath here is the fertile surface of the mushroom, and those are tubes under there, and it's a white surface. Quite beautiful. Now, we can break fungi down into a few different categories as far as their ecological roles. Like the image before, and this organism here, this Coprinus genus, they are decomposers and recyclers. We can call them recyclers, I suppose. They are called saprotrophs, or they have a saprophytic lifestyle, meaning that they obtain their nutrients from dead organisms. So the red-belted polypore is feeding off the that tree, its components, the carbon-based components, and also this Coprinus. And these organisms, at least in a temperate rainforest, we could say, are really critical in unlocking carbon. And they're also critical for replenishing carbon dioxide into our atmosphere. When we think about these organisms, the saprotrophs, the decomposers, we really have to think about their function. And recently I had someone say to me, but wait, don't we want less carbon dioxide in our atmosphere? Isn't climate change fueled by an excess of carbon dioxide or carbon-based atmospheric molecules? And the short answer to that is yes, that's true. And then it's a little more complicated than that as well, because we need carbon-based molecules in our atmosphere. You know, for instance, that's what plants use. They metabolize CO2, and then they release oxygen through their photosynthetic process. That's the byproduct, the gaseous byproduct of photosynthesis. So there's a balance that needs to be maintained there, as well as all of the carbon-based molecules that life on Earth requires. So let's hop to this figure here, that in a temperate forest, there's approximately 2 tons of carbon per hectare. A hectare is, well, actually there's 2.47 acres per hectare. So kind of imagine that, 2.47 acres. And a quarter of that is trapped, so to speak, in the form of woody plant material. And fungi are especially successful at allocating that and breaking that down. We'll spend a much deeper dive into saprotrophs in Unit 2. But just understand that their role is quite specific, and they're incredibly powerful decomposers. Not many organisms can pull off what they're doing as far as decomposing woody material. And the reason this carbon is so important on a terrestrial level is that all of the life we understand on Earth, or what we know as biological life, is carbon-based. And the carbon-based molecules that all organisms use or utilize in some form or fashion um, are lipids, proteins, carbohydrates, and nucleic acids. All of those are built out of carbon molecules. So if we weren't able to access, or let's say the fungi weren't able to access and disassemble those woody materials, a quarter of that carbon in those 2 tons per hectare would be stuck. And that's a limiting resource in the cycle of carbon. So they play an incredibly important role in decomposing. Of course, they decompose other things too. And we're focusing right on this woody material for the brief second. Like I said, we'll go deeper into the decomposers in Unit 2. Here we have a picture of Flammulina velutipes. This is the wild version of a mushroom called enoki. And enoki is an edible. Of course, this wild version is as well. And it's quite prized in Asian cuisines. And it's pretty delicious. It looks entirely different. The form that the cultivated version takes is a long stalk with a tiny little button stem. If you look up a picture, you'll think, wow, they don't even look the same. They're actually grown in constricted environments and harvested at a particular time in their growth phase so that they keep that long spindly shape. And this one you all may know of or may recognize, turkey tail, Trametes versicolor, another decomposer, another saprobe. And then another couple of groupings that we can put fungi in their ecological roles are as parasites and mutualistic symbionts. Now, symbiont refers to the term symbiosis. And I imagine many of you are familiar with that term, symbiosis. And it's often associated with a positive interaction between species. But what the term symbiosis means, strictly, is a deeply interconnected relationship between organisms, between species. And in that term symbiosis falls a few categories: mutualism, parasitism, and commensalism. Now, commensalism is a condition where both of the organisms are okay. They're at a state where no one is really losing anything, and it's a fairly benign situation. Mutualism, both organisms are benefiting in this symbiotic relationship. And as far as fungi go, we see that most specifically in what we call mycorrhizal fungi. Myco referring to the fungus, and rhizae referring to the root. And what that mycorrhizal relationship is there is a fungus. Many species do this. The fungus infects the root system of a vascular plant, and those two individuals begin sharing nutrients with each other. The fungus will reach out into the substrate further than the roots can, and at a far greater surface area than the roots can, to access nitrogen, phosphorus, and a bunch of other minerals, and in a way trades those for sugars from the tree. So the tree or the vascular plant is receiving all of these nutrients from the fungus. At the interface, the fungus is receiving sugars that the tree or vascular plant is synthesizing through photosynthesis. It's a really, really fascinating situation and quite critical. Right around 98% of vascular plants have this relationship with fungi. There are plants that really need this fungal relationship, and we also see that vascular plants do pretty poorly when they don't have this relationship intact. Again, we'll go deeper into that, and we'll jump into that one in Unit 3. I want to give wide berth and a lot of space to talk about that relationship because it is so important and so critical and really, really fascinating. And then the other ecological role that we find fungi in are as parasites. And that is a situation where one organism benefits from the relationship at a detriment to the host. And sometimes the parasite kills the host, and sometimes it doesn't. Sometimes it takes a long time. That's something we see quite a bit of. And interestingly enough, there's a handful of organisms that are parasites on woody trees, and then we'll also step into the ecological role as a decomposer. Once that tree dies, it will then consume its dead body. So that's an interesting relationship there, and we'll dive into that in the next unit, in Unit 2. Well, morphology is the shape or the form that an organism takes on, and it's the study of those features. It's the study of, how the cap looks, how the stem looks, etc. That's what morphology is. And this section is diving into the morphology, the way that these fruiting bodies present themselves as features to identify with. And this is a really incredibly important process to get a hold of. And each of these components that we're going to run through are important for identification. There's a lot of new terms in here for us all. There's a lot of generalizations that you'll see, and unless you have a context of looking at, for instance, a gill from this mushroom, a gill from this mushroom, a gill from this mushroom, they may all seem kind of the same to you. But with practice and observation, you'll be able to get pretty good at this. And you'll be able to use this to do field identification. And a great goal to have is to be able to ID a mushroom to its genus by their visual characteristics or their morphology. And that's one of the goals for this course that I have for all of you. So the first thing that we'll jump into is the cap. And the cap is also called the pileus. You can refer to it either one. If you want to get fancy, you could call it pileus. But the cap provides the structure for spore making, so to speak, the reproductive structures and the fertile structures. It's the container, and it also creates a wind current, a micro wind current around the cap, whereby when the spores are ejected from the cap, from below the cap as it opens up, the air current kind of is faster along the surface of that mushroom cap, and it propels the spores a little further. Here we have a picture of a very, very young Amanita muscaria. And this mushroom is just coming up, and its cap hasn't opened yet. These caps become very broad and flat, something we can call convex or planar. And I also want you to notice some of the features on the top of that cap, and the colors too. Those are important. Here we have a picture of a mycena, and mycena is a very small mushroom, very beautiful. And some of the features that we're looking for with the caps, you can see these two mushrooms are entirely different. Have a look at that. Very different fruiting bodies. Pretty easy to distinguish the two of them apart. But some of the finer details, like for instance when we step into the genus Mycena, there's a few that kind of look like this one. And some of the cap features here will help us identify this to the species. And what we're looking at is the cap shape, whether it's broad or flat, convex, umbinate, and I can point out something right here. There's a little kind of like a gentle curve there. There's really not something that looks like a nipple, which sometimes occurs, and it's not convex or concave, I'm sorry. Umbinate refers to that feature of being concave right at the center of that cap. And of course they can take on all different shapes and features here. We're also looking at the cap surface, and we want to see if it has any features like fibers on it, if there's scales, if it's warty. That image before of the Amanita is described as warty. So you might find that in guidebooks as a descriptive feature. And here's something radially striate. What that is, you can see these lines moving around the cap, radiating from the center. These are called striations, and they come in different forms. This is a striation that Mycena will present. And of course the texture of the cap, as I said before, if there's any surface scales or warding, also the cap margin or the edge of the cap is important to look at as well, and that could be smooth, it again could be striate, it could be undulating, there could be pieces of fiber hanging off the edge, and we'll talk about what that fiber is in a little bit. And there are features of the stem that we want to talk about as well. The stem is also called a stipe, and the function of the stem is to raise up the reproductive components of the fruiting body, i.e. the gills or the tubes or however those spores are ejected, to raise it up above the substrate it's growing on to catch a higher air current so that those spores will travel further. And these organisms present in many, many different ways. Their morphology is quite distinct sometimes. Here we have a Pholiota scorosoides. This is a wood decay mushroom, and you can see those stems are really unique. Now the caps haven't fully opened yet. These are young specimens, but the stems are really shaggy. They've got like these scales all over them, and thinking of the cap, have a look at those caps right there. You can tell from this picture that they're slimy, and they generally will maintain that slime or texture, that stickiness. It's like a tacky goo. They'll maintain that throughout most of the age of their life. Of course, unless it gets really, really dry in the environment, they'll just dry up. But those will eventually open up. And some of the descriptive features of the stem or the stipe is that they could be smooth, they could be reticulated with what looks like spider web features that won't brush off, fibrillose, they could be twisted, they could be equal, they could be hairy, they could be fuzzy, and there's distinct words that describe that, and you'll see them on mushroom annotation sheets and the in the guides that you look through to identify mushrooms. Some guides will be a little less mycologically verbose, and others will be a little more towards the layperson where those words won't be used as often. And of course, the shape of the stem is important. Believe it or not, they could be skinny at the top, tapered at the apex, and then bulbous at the base, or vice versa. And caps also have a different texture, or I'm sorry, stems also have different textures between all of them. They could be hollow, they could be flat, and they could also be kind of dense. They run the gamut. Now, this is a really important thing to look for, especially for those of you who are interested in eating mushrooms. And the reason this is important is because, well, of the genus that creates these structures, remnants of the universal veil and the partial veil, which we'll get to in a second, those organisms, in those genera, there's a few species that are very, very deadly if consumed. So have a look at the center of this picture here, and if we look down at the bottom, we can see this, what looks like a little egg with the shape of a mushroom in it. Well, that is called an egg, and this is the beginning of the growth of this fruiting body. This is a common name for this mushroom is Caesar's mushroom, Amanita jacksonii. And as this organism grows, oh, I lost my mouse somewhere, there it is. As this organism grows, it will pop out of this egg. As we see, it will continue growing and grow and fully open up. There's 4 different ages of mushrooms here. Now, what's unique about this is the remnants of this egg are left on all of the older specimens. So when you find this in the forest, in the leaf litter growing under trees, you will find this cup, or this, it's called a vulva also. And these are remnants of what's called the universal veil. And the universal veil is that covering in which that fruiting body grows from. Here is a picture of another Amanita species, and we can just begin to see, it's a little washed out here with the light, you can see that at the bottom of this stem, the bottom of the stipe at the base, is a cup. Now, when I approached this mushroom, it looked like this. It was coming up out of the detritus, the leaf litter, the sticks and the needles, and it wasn't really apparent what was happening down there. So when we're picking a mushroom for identification, or even not picking it, even if you just want to look at it, it's important to clear away some of this detritus to see what's happening down there. And of course, below that mushroom is a whole network of mycelium reaching out into the soil. Amanita are mycorrhizal, by the way. So these two species are mycorrhizal, and they're connected to the vascular plants in their forests. Here we have another Amanita, Amanita rubescens, the blusher it's called. It's a really beautiful mushroom. Below these leaves is the cup, remnants of the universal veil. But there's something else here that you'll notice on the stem. A feature of the stem that we really want to look out for as well is this, what's called a ring or a veil. This is a remnant of what's called the partial veil, also called an annulus, by the way, this ring. This partial veil covers the gills as they develop. So before slugs got to this little guy, the gills were protected by a partial veil under here. And as the mushroom grows and opens up, as the cap opens, that veil will tear off. And what happens with certain species and certain genus as well, that veil won't dissipate. And it will either form something like here in this species that's like a draping, it looks like a cape or something, it's draping. Some rings or some annulus will actually detach from the cap or from the stem and create a ring that can slide up and down the stem. Or sometimes what's left is not even anything this apparent. You'll just get like these little bits and pieces of what used to be the partial veil, and also things that look like spider webs stuck around the stem, which is an indication of a genus also. Something else that will happen, we talked about the cap margin, and I said they can sometimes have hanging bits. Well, sometimes those hanging bits are left over from this partial veil. So this partial veil can break off and remain persistent along the cap edge, the margin, as well as the stem. Here's an example. The remnants of the partial veil of this particular mushroom are really different than the last one, right? It looks like a fuzzy, a fuzzy mess. But as that mushroom opens up, it will be persistent and is a key identifying characteristic that we can use. And of course, we want to look at the gills of the mushroom. If we have something that looks like this fruiting body, we call these blade-like gills. And how they look and present themselves is really important for us. And of course, they're the location of spore production. This is the fertile surface of the fruiting body, also called the hymenium. This is where all of inside here, microscopically, if we were to take a cross-section, we would see images of those basidia, those structures that grow spores and then eject them from the surface. Gills are also called lamellae. I probably won't say that too often. It might slip out here and there, just so you know. And some of the descriptive features that we're looking for, we'll start with the shape and the thickness. We'll kind of go backwards here. The shape and the thickness, like, is important. Like, these look like thin blade-like gills. You can see that they're fragile. Slugs have been eating on this, and this mushroom was kicked over by accident. And in that, really, it was a kick that was like just shuffling feet. It damaged all the gills. This is a mushroom called Stropharia rugose annulata, and the common name that you may be familiar with is wine cap. These are an easily cultivated mushroom Tastes delicious too. Um, but these gills, we're looking at the color. They're darkly colored. These are mature as well. So as they age, they change color. So a young mushroom will have lighter colored gills, at least in this species. And then they'll age to like a purple and then almost to a black. Not all mushrooms will do that. Some fruiting bodies will maintain a very similar color over time for their gills. And also, we want to see how close they are to each other. And that's one of those relative things. If you don't know what a close gills look like versus broad gills or subdistant, it doesn't mean too much to you. But we can see it looks like there's some space in there. I think without having looked at many mushrooms, you could say that it looks like there's some space. They're not crowded, but they are close. And one of the other super important features we want to look for, and this is recommended for every mushroom you identify, is to look for its relationship to the stem. That will tell us a lot about this fruiting body and who it belongs to. That could be everywhere from completely touching the stem. Like this is an example of a gill that reaches all the way to the stem, and if we were to zoom in there, we would see that it's touching. And it also, in other fungi, they could be free gills where the stem never even, or the gill never touches or reaches the stem in their growth pattern. This image right here will be a little more clear for you. This is the annotation sheet, or the annotation sheet for gilled mushrooms, and I think this is really useful to hold on to as we go on through the rest of this. If we look at the gill attachment right up here, we can see that we can visually see what these definitions mean. And these are all cross sections of caps, so we can see the gills. There's my mouse. I think that you all might be seeing my mouse the whole time, and on my end I can't really see it until it's always moving. So please be patient with my roaming mouse cursor. So here we have an image of a free gill attachment. You can see that this blade of the gill doesn't really touch the stem. And here we can see a nearly attached form. It's called adnext, where it's just touching it. Adnate, where it's straight across. This one has a bit of a tooth, so it's starting to come down the stem a little bit. This one has a little sinus, we call it sinuate, a sinus before it actually reaches the stem. And decurrent is something you'll hear a lot of. There's some edible species that have decurrent gills. And ecruate. I don't hear this one very often in books, but decurrent, I think that ecruate and decurrent might get lumped together sometimes. So those are the images of what those definitions are. This sheet is super useful. Even when you're out in the field, I like to bring one of these with me and just kind of keep it with me to keep me on track with some of the features that I should be looking for. We won't go into this sheet together, but please use this through this this slideshow to keep yourself, having visuals, because I can't quite fit all of that in there. It would be a very long presentation if I did that. Here is an image of some gills that we can look at up close. This is Pholiota squarosoides, and this is the gill pattern of this fruiting body. And we can see that the gills, I would say they're close but not crowded. We can see they're a little bit yellow. The lighting, I don't know, they might be a little smoky looking too. That could be a descriptive of the color. And the gills, if we look, they run all the way to the stem. They are attached, and sometimes you have to get pretty close. The strict definition for this is bluntly attached. So another thing to note is that occasionally you'll find gills that in a mushroom that's supposed to have like bluntly attached gills, for instance. Every once in a while you'll find one that doesn't reach the stem. It's not often. Like there will be no question if you took the gross average of how all of these gills are approaching the stem, that will be your answer, whether they're attached or not. And it does take some practice to see in the field what free, what attached is, what adnate is. It takes a little bit of practice, but that is a feature that we always want to look at. Here is an example of a different gill pattern. Now, the previous images, we were looking at blade-like gills. Now these, right here, on the sides of this scaly vase mushroom, which is Turbinellus flacosus, is these really aren't blade-like gills, are they? They're more like ridges. If you were to see this up close, you would see that they're more akin to folds. And that is all the fertile surface. That's where the spores are produced and ejected. And they run down the whole length of the fruiting body. A little bit down in here they kind of dissipate, but these are certainly decurrent to an extreme form, and they're presenting not as blades but as folds. And the light shining through here, and it kind of gives you the idea that this is like a vase. Probably right down in here there's a concavity, giving it the name scaly vase. Here is a Lactarius species, and Lactarius rufus is, I think one of the common names is the hot mushroom, or the spicy mushroom, because that little latex here is very, very spicy to taste. We'll get back to what you can taste and what you cannot taste in our section, Can You Eat It? It's generally not recommended to taste a mushroom if you don't have a lot of experience with identifying species. There are some genera that you can get away with putting in your mouth and just kind of getting a taste and spitting it out. For our purposes here, I'm going to recommend against that. But what I do want to point out is that these gills here, when they're disturbed or broken or cracked, and they're very fragile, they exude a latex, sometimes referred to as a milk. And the features of that latex are important. Well, first of all, the very fact that that happens lets you know that it's a Lactarius. That's an easy ID right to a genus. And sometimes that latex will stay white, sometimes it will turn clear, sometimes it will turn black, and that's a key identifying characteristic among the Lactarius. And of course we can see these gills, check that out. They're starting to reach down the stem, slightly decurrent, might be called subdecurrent. I'm not sure what the guidebooks say, but that's pretty characteristic of this. Of course, we can have organisms that really don't fit into what we've talked about yet at all. And this is a mushroom called I know it as the hedgehog mushroom, as a common name. There's a couple of others. I almost think one is called the sweetbread mushroom, or maybe I'm misspeaking there. I think that might be a toxic mushroom, believe it or not. But this is Hydnum repandum. And actually, backing up a little, I try not to use common names often because common names get bumped around between different species and it can be confusing. Like one person could call the tinderconch, call Fomus fomentarius the tinderconch, while other people call Ainonotus obliquus the tinderconch. And then there's a couple others that are called the tinderconch as well, and it just gets confusing. So I usually stick to genus and species when I'm talking about identification. So here we have Hydnum repandum, and they present, instead of blade-like gills or folds on the undercap surface in the fertile area, the fertile surface is actually these little, what are called tooths, or tooths. It's called a toothed mushroom, and they're like stalactites. And then all along the surface of these stalactite-like teeth are basidia producing spores. And we can also have fruiting bodies with tubes rather than gills or folds or teeth. And these fruiting bodies fall into an overall term called a bolete. That could be a little confusing because boletes are also a genus, and within that broad term bolete there could be a lot of different genera, a different genus, and basically, boletes, the broad term, falls into like fleshy stalked mushrooms that have pores, like this one here. Also bracket fungi, like artist conch, that's Ganoderma applanatum, Fomus fomentarius that we saw earlier with Utsi, Piptoporus betulinum. They also have pores instead of gills. And that is the production space for the spores. And they can come in a few different shapes. They could be circular upon examination, or they could be angular as well. And if we look in here, we can see that these are angular pores. And this is the Boletinellus marioloides, beautiful mushroom. I think we're going to talk about that one a little bit in Unit 3. There's some interesting symbiotic relationships in there with this organism. And this one, Swillus spraguei. Now, if we look closely, we can see that those are also angular pores, and all within that surface are spores that are being produced. What's interesting about this mushroom is that I found it up in a tree. It grows on the ground. It grows in the soil, and I found it up in a tree, and this is a remnant of what squirrels will do. They will pick mushrooms and hang them up in a tree or in the crook of a branch, and essentially store them for when they're ready to eat them. And they can even dry out completely, and they'll come back to it and feed on it every now and again. So if you're paying attention while you're out there in the forest, you might find some fruiting bodies in trees. That is the work of squirrels, likely, and other organisms do that too, but squirrels mostly. And here we have a picture of a microscopic view of spores from Coprinus comatus. A common name of Coprinus comatus is shaggy mane. It's quite a delicious edible. And Cuprinus is also broadly called the inky cap, that genus, because their caps kind of dissolve away in a process called deliquescing. But here we have images of spores. Here's the basidia that the spores are produced in and grown on, or grown from, I should say. And then here they are, all atop the basidia. Now, spores are the mechanism for genetic dispersal. So we can think of these kind of like seeds, but this is how fungi reproduce sexually. And in some occasions, actually, I think all of these sexually produced spores can be ID'd microscopically. So if you wanted to take it to the next level, you could get a very cool guide for microscopic spore identification, and then jump into that world and start to ID by features of the spores, size, shape, etc. It's really interesting. And something that we should always be doing with identifying fruiting bodies is taking a spore print. And as you get more comfortable with IDing mushrooms and fruiting bodies, you might want to collect them for food or for medicine. I highly recommend that whenever you plan on doing something where you'll be consuming that fruiting body, you have to have a spore print, because that will oftentimes be the defining feature of identifying that fruiting body, that mushroom. And I know a lot of people that have been saved from eating a toxic mushroom by waiting and pausing to take the time to do a spore print. And what a spore print is essentially taking the cap off from the stem of the fruiting body and placing it down on a sheet of paper, gill side down or tube side down. And something that I do, so these are two images of dark spored mushrooms. We also have very light spored mushrooms, even white spored mushrooms. So if you lay one of those caps on top of a white sheet of paper, you actually won't get to really see the spores, or you might not really get the subtle changes in shade or tone. So what I do is I will sometimes I'll draw a circle about the size of the cap, and then I'll color half completely black. I would color this side all black with a Sharpie marker, fill it in all the way, and then take that cap and lay it down on top of that circle that I've drawn where I have a white side and a black side, and allow that cap to sit for some time. It could be over the course of an hour that it takes to get spores to eject out of the cap onto the paper, or it could take a couple of days before spores start collecting. It depends on the age of the mushroom and whether it has spores to release. And here's a collection of images. We can see there's a lot of different colors, there's a lot of different shapes. Really, really important feature here. So let's come back to this. When you're taking a spore print, you want to put these mushroom caps and these fruiting bodies out of the reach of small children or pets. For some reason, every once in a while, a pet will eat a mushroom, or a child will eat a mushroom. And if that's a toxic mushroom, that would be a really horrible outcome. And we can think of these as potentially very dangerous items to have in the home. If it's yourself, you don't have to worry about it. I don't think you'll accidentally eat one of these. But I recommend keeping them at a distance or in a place where they're safe from being accidentally eaten by a young person or an animal or a pet. Now, one thing that will help releasing these spores is that if you put a cup or a bowl over the top of the mushroom cap as it's laying there on the sheet of paper. And the reason that makes a difference and is especially important for very tiny, tiny mushrooms. So there's two things that happen. If we have a very small mushroom, like think back to that mycena, if you can, sitting on top of that log in the photo on the moss, that's where that was. Mycena are a pretty small cap. They could be like maybe 5 centimeters or 3 centimeters even. They're pretty tiny. If we were to leave that out in our house, it'll dry up pretty quickly. It'll desiccate. And when that happens, sometimes the mushroom cap won't release spores properly, or as much as we'd like, I should say. So by putting a cup over the top of that, or a bowl, or a glass jar, you're keeping moisture in that microenvironment for that cap to release spores. Now, it also prevents there from being wind currents along the paper and along the cap. Now remember back when we were talking about features of the cap, one of the functions of a mushroom cap is to create wind currents, micro air currents along that cap surface to carry spores far away. Now, we want the spores to land directly underneath the cap. So we want to take away that functioning of the cap. So by putting a jar or a bowl or a glass over the top of a mushroom cap, when we're taking a spore print, we're taking away that air current so that when those spores are ejected, they fall right down onto the paper, and it'll be less time before we'll need to, or I'm sorry, it'll take less time to collect those spores so that we can get an accurate look at what color those spores are. So I may not have mentioned this. The reason we're looking at spores is to check our guidebooks to see what color the spores are for these organisms. So all of the spores have different shades and hues. Some are white, some are black, some are brown, some are rust brown, some are purple black. Really a wide range. There's even green spores. So we're looking at the color of the spores. Look at that table of all those mushrooms. It's a beautiful sight. This was an especially fun class here a few years ago, a mushroom apprenticeship that I was teaching, and we can see that we've collected quite a few specimens, and they are all different shapes and all different sizes. So we're going to jump into collection protocol. There is a process to this. I think you're getting the idea, as we've gone through morphology. Those are all things we want to record and pay attention to as we're collecting. And here's some tips to make that a good and successful process for us all. So we'll want to have a container. And the best container is a rigid container that will keep its shape, so when you put delicate mushrooms into it, the mushrooms won't get crushed by each other. I like to use a flat-bottomed, rectangular, or square, or even round basket. If you don't have one of those, You could use a paper bag, or you can use wax paper to kind of fold those mushrooms into. And the reason we don't use plastic is because plastic doesn't breathe, and depending on the weather and depending on the species you're collecting, if you have a mushroom in a plastic bag, it just can turn to mush within the matter of 15 minutes. And they're not really great for storing fruiting bodies. What also works really well is a laundry bag, a mesh laundry bag. There's a lot of airflow through there, so the mushrooms won't start to break down in the hot sun or with humidity. And another feature of a basket and a laundry bag is that they're open, and we can continue to spread the spores of these organisms. As you'll see when we get deeper and deeper and deeper into these ecological roles, we want these organisms everywhere. Mostly. Maybe foresters don't want them around if they're decomposers or loggers. But we want to spread these organisms across our landscape. We want fungi to be successful because our relationship is and survival is tied intimately with these organisms. So when we have a basket or a laundry bag, we're also spreading those spores and ensuring their success and ultimately our success. So I like a basket. I think they're great. And also if you have like the right handle on it, you can just like hang it in the crook of your elbow, and it's like you don't even have a basket. You're just kind of rolling along and checking things out and popping mushrooms in your basket. Now, the other thing we'll want to have is a notebook and a pencil or a pen. I prefer pencil because you can erase things. It's important to have those because we're recording features that we're observing. We'll get to that in a second. A hand lens is very useful, or a magnifying glass, because you can have a look at the fruiting bodies up close, and there are features like if there's a remnant of the partial veil, remember that annulus hanging down in that Amanita picture? Sometimes the remnants of that partial veil are very, very minuscule, like fibers. And it would be hard to see some of these features without a hand lens. And if we have a hand lens or a microscope or a magnifying glass, we can look up close in the field, right where we've collected this mushroom, to have a look at it. And the reason it's important to do it in the field as well is that these fruiting bodies will change over time. As soon as we pick a mushroom, it's lost its water source. And that's pretty critical for very small fruiting bodies. And we want to be able to see all of those features right quick and record them. So a hand lens is really nice to have. Of course, cameras are great too. A lot of times when I don't have a pen and paper, pencil and notebook, I don't have a container, or I'm in a hurry, let's say I'm backpacking and I'm not actually out identifying mushrooms, or my head's in an entirely different place, I'll take a picture of a fruiting body and I'll make sure that I get the picture of the fruiting body as they're standing, and then I'll pick a mushroom and tip it over so that the gills are showing, and I'll take a picture of that standing fruiting body next to a mushroom that's tipped over so I can see the gills and the gill attachment and some of the other features. And you can record a lot of details just with a camera or maybe your smartphone. That tends to be my go-to just because it's in my pocket, unfortunately, often. And a knife might be useful too. I would say a knife is useful for cutting a mushroom in half to help see how the gills attach. I think that's really the only time I would use a knife. Once in a while, if I want to collect a bracket fungi or a woody fungus that's on a tree. If I want to collect one for a species, or for a voucher specimen to take home to identify, I'll use a knife to help get that off the tree, but usually you don't need one. Good to have if you want to do some field ID of gills, but not entirely necessary. And of course you want a snack and water when you're out in the woods. Don't forget to take care of yourself. Now, some of the things that we're observing, believe it or not, the weather conditions are important. We just had here a stretch of very, very hot and dry weather for about 3 weeks, and then had 4 days of spotty rain, and now the mushrooms are back up. Prior to that, there wasn't a whole lot happening. So recording the weather conditions will help us understand the growth habits of certain species of fungi. Some are less sensitive to water decline or less sensitive to drought than others. And some mushrooms will pop up within the matter of a couple hours after a hard rain. So it's nice to be aware of how those organisms are producing fruiting bodies. And of course, if you put the date down, you'll know when to find these mushrooms. Whether it's something you like to forage for food or for medicine, or you're keeping a tally of how mushrooms are responding to global climate change, or the adjustments, or I guess the changes to our atmospheres and temperatures and rainfalls. It's a very complicated system, and I think we might be able to use the frequency of some of these organisms as indicators as to what's happening in our temperate forests. Another very, very important feature to look for, something we haven't talked about yet, is the substrate on which the fruiting body is growing. And it'll be, in the forest, it's often growing on wood, or it's growing on the ground. On wood, it could be a hardwood, a deciduous tree, or a softwood, like a conifer. It could be a highly decayed piece of wood, or a highly decayed tree, to a tree that is even still alive. It's important to record the quality of the substrate here. That will give us an indication of who this organism might be. And also, if it's growing on the ground, we can look at some of the features, like whether it's cultivated soil, whether it's sandy soil, whether it's disturbed soil, whether it's a parking lot, a hard-packed dirt parking lot, versus a grassy patch in the forest. We also want to look at the plant species that are around. If you can ID the trees nearby, that's awesome, and you should. Especially if it's growing on wood, you should try and figure out what that tree is. That'll give us some ideas as to who this might be. And a lot of times we can identify down to a genus, even a species, based on what it's growing on. And I've had conversations with several people, some people that I know that are pretty good foragers. Starting out, they've almost eaten poisonous or toxic mushrooms without paying attention to what the substrate of that mushroom was. For instance, I'll share this much. Chanterelles, orange mushroom with ridges, and we'll talk about these a little bit later. Chanterelles grow on the ground. No doubt about it, they grow on the ground. There's another mushroom, the jack-o'-lantern mushroom it's called, it's a common name. It grows specifically on wood and has blade-like gills that are decurrent. The chanterelle has ridge-like gills, or ridge-like folds, that are decurrent. if you saw these two things together, you would never think that they looked similar, but they're both orange. And believe it or not, a lot of people accidentally eat the jack-o'-lantern thinking that they're chanterelles. I think it's probably a beginner's mistake, but one of the features that would give it right away, if for some reason you're not paying attention to the gill differences, one thing that will give it away is jack-o'-lanterns grow on wood and chanterelles grow in the soil. On occasion, we'll find fruiting bodies that are growing what seems to be out of the soil, but they're really growing out of a stump that's underground or a log that's under soil, and it's emerging from that. So sometimes we have to dig down a little ways to see what that fruiting body is really growing out of. So I like to think about being out in the woods as kind of like identification missions for fungi or fungi. And you know, some people will go out with the mission like, oh, I'm gonna go get turkey tail, or I'm gonna go find morels, or I'm going to go find chanterelles. That's entirely fine, and you know, it's a great way to get out and observe the natural world with a focus on an activity. But for this class, I would like you all to kind of temper that excitement to find a specific species for now, and let our curiosity guide us. Go out with, you know, the eyes of a child and take in these organisms as they are, as purely mysterious things that just pop up sometimes overnight. Take in the colors, take in the smells, take in all of the features of the natural world, and just go out and have a good time. Like, go for a walk. One of the really cool things about mushrooming or going out on a foray is that you typically don't go too far before you find a bunch of things to get really excited about. You're like, oh my gosh, what is this thing? What is that thing? Oh, look at this, that's growing like this. And if you're out with people, it's even more exciting because, you know, 2 or 3 of you are out in the woods, or even 10 of us, we'll be out strolling around, and we'll end up, we could go 50 yards in 3 hours even. Very, very short distances to find all sorts of cool things. And of course, you know, bring your camera, your drawing pad, pencil. And if you can draw pictures of mushrooms, I highly suggest it. When we draw something, even if you're not an artist, and you don't have to be an artist to do this, when you draw something, you're really, really closely looking at it. You're looking at things you've never seen before at first glance. You're looking at how colors and light interplay off each other. And if any of you are artists, you'll know that when you're adding color to a piece of work, whether it's watercolor, oils, pastels, there are colors you would never expect to be on a plant. Purples and blacks, or in a mushroom. The range of colors are even more spectacular. So drawing a fruiting body is a great way to get to know it, to really build your relationship with that. And the very last thing I'll say is pay attention to where you are when you're out in the woods. It's very, very easy to get confused and turned around and even lost if you're looking at the ground for mushrooms. It's so easy. I consider myself to be a very direction-oriented person, and I can travel all sorts of places without fear of becoming lost, you know, paying attention to the direction of the sun, the topography, etc. And even still, if I'm focused on the ground looking at mushrooms, I can get confused. So pay attention to where you are. A great way to start out actually is on a trail and following this trail and examining what's just off the trail. Unless, of course, you're very familiar with a patch of woods or a patch of forest. Have fun, go for it, go explore. But, be careful, don't get lost. And some things to really look for when we're doing this, of course, we're looking for the substrate, the trees, the forest type, the tree species. We can collect individual fruiting bodies also into our basket with wax paper or individual paper bags or sandwich bags. We can keep all of those mushroom fruiting bodies separate from each other and cataloged really well in our basket as we're out exploring and collecting. Also, you can write the key identifying characteristics that you're noticing in the field right on the paper bag or the waxed paper or the sandwich bag before the mushroom goes into the bag. And that's a great way of keeping information attached with the fruiting body. Because if you put a bunch of mushrooms into a basket and then have a bunch of notes, if you have a lot of them, it might be confusing when you start to connect the written information with the specimens you've collected. Also, collect high-quality specimens for identification. Typically what I will do is I will choose a specimen that is large enough and, well, I should say mature enough that I suspect it has and is producing spores. And that will be my specimen for at home doing a spore print. I wouldn't choose anything young, like we can look in this picture right here. Wow, this Foliota keeps coming up in this presentation. Must be a cool mushroom. This Foliota, you can see right here underneath this cap and underneath this cap, they're a little different. This one's opening up, and this one is still closed up and has remnants of the partial veil intact. And you can see remnants of the partial veil here on the edge of the cap at the margin. But here it's mostly covering up the gills, and definitely here that partial veil is still covering up the gills. So I wouldn't collect any of these young ones for a specimen or for a voucher species. Or a voucher specimen, I should say. If I had to pick one, I would choose this one. Hopefully the spores are ready in that. I don't know. It does look a little young to me, though. It might not have spores ready yet at this age. So the bottom line, and we'll jump into this when we talk more about edibility and medicinals, but we can apply this to collecting specimens for ID as well. Take only what you need and leave the rest. And for a specimen, like to take home to ID, no more than 2 fruiting bodies. A lot of times we only find 1 mushroom, and therein lies the rub of ethical harvest. If we're collecting something for ID, is it okay to take the one and only mushroom that's out there in the field, or should we leave it? And I typically stick with a 10% rule when I'm harvesting mushrooms and plants for that matter, 10% of a viable population for food and medicine. When there's one fruiting body and you want to ID it, that falls into a different category of understanding. And we have to figure that one out on ourselves, I think. Oftentimes, if I find that situation for myself, I won't pick the one mushroom. I'll try an idea on the spot, or maybe I'll use my camera to get all of those macro features, those key identifying characteristics out in the field, and then use my images to get close to what I think it is. And then if I can, I'll go back to see if a few more have popped up. Usually when you find one mushroom, there's a bunch more, You just got to look around. So you could be patient in return, or if you find only one mushroom, spend a little time in that area looking around for another example of that species, and then you can use that for a specimen to take home and examine. So that leaves us, folks, at the end of Unit 1. Thank you all for tuning in and hanging out with me as we go through some of these details. I hope that you're able to get out and explore the outdoors a little bit and look for some fungi. And we'll be catching up online shortly. And Unit 2 is coming up in a couple of weeks. We will cover the good, the bad, and the ugly of fungi. That should be promises to be a really interesting presentation. There's a lot of really interesting organisms and super crazy things that fungi do. In the meantime, enjoy your forays in exploring. One thing to know, and this comes up a lot in conversations that I have with people, is that in North America, if you pick a mushroom, it will not make you ill. You won't have dermatitis. You won't get poisoned by just touching it. You have to eat it. You have to consume it. So there's a level of fear when interacting with fungi and fruiting bodies, because there are some deadly toxic mushrooms out there. But just know that you have to consume those fruiting bodies in order to be injured by them. So just be careful. Don't accidentally get mushroom parts on your hands and eat a sandwich. Just make sure that you keep your food away from your mushrooms and your mushrooms away from your kids and dogs, and everything will be fine.